Yeah, okay. Hello, everyone. Thank you very much for coming. Maybe I'll leave the door open because it locks or something, and then I'll close it. It's a great pleasure to see so many people here, and thanks for those who have joined us online as well. For those of you who are joining us online, there's about 700 people in the room here, and my name's Donald Trump. No, this is the first invited seminar that we're giving for our IRTG on accelerating crop genetic gain. And it's a great pleasure to have Greg Grabetsky here. Greg is known to quite a number of people from his long career as a plant breeder. I think your official title is you are a Chief Research Scientist at CSIRO in Canberra. I was very disappointed that he didn't wear his headgear with the feathers. But there you go. He was warming up earlier. Some of you probably missed it. I think he's expecting some brutal questions. So make sure you come up with some brutal questions, hard-hitting questions, shall we say. So Greg has worked as a plant breeder for quite a number of years. He is a UQ alumnus. He actually did his undergraduate at Gatton, and then he did a master's on mung bean with a number of people, including Bob Lawn, who would be known to many of you from CSIRO, who has long since retired. He then went to North Carolina State in the US and did a PhD. Greg has served in a number of roles other than being a plant breeder. He's been on the GRDC Western panel in Western Australia to try and help the people in Western Australia to understand what good science might look like. Hopefully you've succeeded in that. That's really good. So Tolera Fufa has raised a hand. What does that mean? So someone at Warwick has raised a hand. It could mean that they can't hear us. Don't know. Tolera, if you've got a message, can you put it in chat and we'll check out what's happening? Thanks. And if you can't hear us, I don't know what you're going to do. And so just to give people background, because I know a number of you who are attending today aren't actually part of the International Research Training Group. So the IRTG is a joint program between UQ and 3 institutions in Germany. It's led by Rod Snowden at the Justus Liebig University in Gießen and myself here at UQ. We have entered into an agreement with the German DFG, which is like the German ARC, to train 48 PhD students. We currently have 50 enrolled, and I think there's going to be more. So I think we will reach our target, which is an important thing for us from the DFG point of view, because in 2027, they will review our program. And if they feel it's been successful, they will give us funding for another 4.5 years. So this, if it is a successful IRTG, and I have every reason to believe it probably will be, it will go till 2032. And we will make accelerating crop genetic gain a sport at the 2032 Olympics and have our final meeting here in Brisbane. Who knows where, but probably not the Gabba. So without further ado, Greg is going to talk to us about breeding systems resilience and not crop resistance for reliable crop production in future climates. And before I pass on to Greg, I'll just acknowledge country. The UQ acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. And over to you, Greg.
I'd like to thank Ian, Jules, Megan for organising this get-together, and it's great to see so many familiar faces, friends, both now and into the future. So thank you. It's nice to be here on sacred land, UQ land. So I undertook my master's. So I'm going to talk a little bit about delivering on genotype by environment by management opportunities. And the focus is really around breeding for systems resilience, reliable crop production. The reason this is important is highlighted nicely by this figure, which shows changes in April cropping rainfall for the period from 1900 to 2000 relative to the period from 2000 to 2021 for April cropping rainfall. And it highlights a fairly grim picture that throughout much of the southern part of Australia, which represents probably 90% of the Australian wheat belt, we expect to see below average or well below average rainfall for this critical part of the cropping season. This has come through for BOM to make the summarise that there is a high degree of confidence that Southern Australia will spend more time in drought in future years, consistent with these projected declines. I'm very interested in climate change and what we as breeders can do to address or improve towards adaptation to climate change. And in doing that, we know that future climates in Australia will be characterised by greater atmospheric CO2, warmer air and soil temperatures, earlier and more intense frost events, and prolonged drought. So quite a nasty set of conditions, quite a nasty set of, I guess, breeding objectives and targets. And when you look at the literature, a number of strategies have been proposed, from breeding small cycles to rapidly build adaptation complexes of genes in response to small shifts in climate, evolutionary breeding, the use or identification and adoption of stress alleles from wild relatives that sit under fairly challenging conditions without the support of a good agronomic system, or trait-based focus to improve tolerance and resistance to heat and drought. However, when you look more closely at a number of these traits and you characterise them in a way that is meaningful to a breeder, a range of traits here, often the value proposition is unknown. What's the value to a breeding company to adopt a long-term breeding activity incorporating new genetics for a range of these traits? The genetic control is somewhat complex. Typically, genetic variability may be hard to identify, and together ease of selection may be quite challenging. So breeders are challenged by the need to adapt and build new genetics into their programs when it's unclear what they should be doing while maintaining improved quality, disease resistance, and yield potential in a competitive market. It appears though future climate impacts on crop growth are quite complex. So Mark Howden talks a lot about we focus on temperature or high temperature or drought, when in fact our future will be characterised by complex interactions between all of these features. And we know from long-term studies that trait-based breeding works well when there is a clear and reliable signal. The genetic correlation for our selection environment, whether that be a glasshouse or a nursery, with our target population environments has to be high. Yet current evidence and predicted evidence suggests that future climate will contain a reliably predictable increase in CO2, which will impact on increases and changes in temperature and water availability. But around that, there'll be significant environmental variability as well, which will challenge the notion of improving adaptation in a uniform and structured way because of the low correlation potentially across seasons and often consecutive seasons, resulting in potential rank changes for traits and genotypes. So the question in breeding remains how much of the forecast change will be predictable across long-term breeding timespans. So we think about Australian agriculture as a legacy of European agriculture from the first arrival of our settlers. Do we need to take our change our way of thinking from 100 years of farming in what were typically reliable grain-fed systems and perhaps move to a system where we allow the growers to take better control and so by breeding and developing around cropping systems that contain crop varieties that are more opportunistic in response to the changing climate rather than resist against climate change, which in my opinion is potentially unlikely given the poor success of breeding for crop resistance to abiotic stresses over the last 50 years. So what do growers do in adapting? Growers are very clever. They have a range of management tools, sowing dates, long fallows, they plant growth regulators, and they can graze if it's dry. So I guess the question for me, given the context of the tools the growers have, what is the G in future? G by change in environment, by system, that enables sowing decisions given farm and environmental conditions and market conditions. So in a situation where we have the knowledge and the growers have the capacity to go play their seasons in advance based on what they know of their starting moisture, their starting nitrogen, but also predictions from BOM as to the rest of the season. So I talk about playing the seasons. I talk about opportunity breeding for greater season flexibility. And I'll talk about 3 sets of traits. One that provides growers with opportunity around establishment, around vegetative growth, and around flowering. So optimizing crop establishment. And I guess this graph shows quite nicely some work from Bonnie Fleur, where she has shown the change in the sowing break for wheat across the Australian wheat belt. And in some cases, this break is up to 6 weeks delayed. And for a grower, this puts incredible pressure in ensuring that they can sow into moisture. If there's not that moisture there, the risk is they will need to dry sow, and there are problems with dry sowing. Furthermore, as growers moving into earlier sowing, it's becoming a more common practice owing to the increasing sizes of farms, growers are needing to plant much earlier. We're finding that soil temperatures are much, much warmer at the soil surface. This is a sand. On a vertisol, the temperatures are even warmer. And so crops perform quite poorly. They establish quite poorly when sown at traditional sowing depths of 3 to 5 centimetres because of the high soil temperatures and often the lack of moisture that we often see with the cooler temperatures at depth. So a number of challenges in successful wheat establishment with changing climates. This graph here just demonstrates some of the concerns. This was from West Australia, August 2017, a full profile, but no surface moisture. The growers dry sowed. This is the stage of the crops. This is now 3 months into the crop. The growers here had to spray out their crops. So 60% of the cost of growing a crop is with sowing. Then you've got to put on top of that the cost of removing that crop and potential erosion risk and waste, a loss of income. So significant risk with dry sowing and sowing shallow. We're seeing declining autumn rainfall. We're seeing increasing soil temperatures. We're seeing, in many places, reduced carbon. So we find that there's much greater furrow infill, which increases the sowing depth. So the key trait that we're focusing on in a systems resilience context is increasing coleoptile length to ensure timely emergence and assured crop establishment with deep sowing. So coleoptile is the shoot that goes from the seed to the soil surface. It's a modified first leaf, anatomically and morphologically. It's just like a first leaf. We know that, and we know that the length of the coleoptile determines how deep seed can be sown. 2 sets of genes affect coleoptile length. There's the larger cell and faster growth genes. These are genes which are associated with more rapid cell growth. And we've done quite a bit of work, genetic studies. This is some GWAS Manhattan plots just showing the position of alleles across the wheat genome associated with faster cell growth. But the other really key set of genes are the dwarfing genes, the genes that reduce plant height in wheat, but also in other cereals as well. And we know that since the 1960s, when the Green Revolution made such a significant contribution globally to assuring food security globally, associated with that reduction in plant height was a significant reduction in the capacity of crops to establish when deep sown. So the coleoptiles were much shorter. And this has been an issue not only when chasing moisture, but also in warmer soils from the early 1960s. A grower said these these dwarfs couldn't push the skin off a rice pudding. And so they're associated with poor establishment. This graph here, or this figure, shows the change in height as you increase the frequency of these Green Revolution Della mutants. And work associated with these near isogenic sets have shown quite clearly that as you increase the frequency of these gibberellic acid insensitive dwarfing genes, we reduce linearly the size of the cells inside plant leaves and coleoptiles. So introduction of reduced height genes is associated with reduction in cell size, which is associated with reduction in coleoptile length. We've been on a bit of a mission globally to identify alternative dwarfing genes from seed banks from all over the world. Many of these genes were identified out of mutation programs arising out of the Cold War. And so they were noted as being reduced in height, but there was very little done with that material. So our role is to bring these genetics into Australia and then characterise and understand their value in an agronomic context. Some work from Mark Ellis shows quite nicely where we've grown sets of these neoisogenic pairs containing either GA responsive or the Green Revolution GA insensitive dwarfing genes, that the reduction in coleoptile length with the GA responsive mutants was much less than with the Green Revolution GA insensitive dwarfing genes because of the impact on cell elongation I showed you earlier. We then worked with this material a bit further and showed with dark growth studies how the increase in coleoptile length with the new alternative dwarfing genes, shown up in the right-hand corner, was associated with a significantly greater coleoptile elongation rate. Again, tightly associated to the GA biosynthesis and its use and the impact on cell elongation. The gene that we've probably put the most effort in is RHT18. It's a gene that it's a GA2 oxidase, which we identified in some mutant durum wheats in Italy. And this gene has now been well characterized and cloned. We have great markers, and we've been moving these genes into a range of commercial backgrounds through back crossing and then delivering these to breeding programs. Here are 4 examples showing the commercial variety on the left, containing RHT1 or RHT2 Green Revolution, and the near isogenic RHT18 dwarfing gene, sister line to the right. Our first look at these pairs, the 18 represents the RHT18 near isoline, showed a significant reduction in coleoptile length when we maintained RHT1 or RHT2. And that by replacing these genes with RHT18 through greater coleoptile elongation, we also increased coleoptile length significantly. And on balance, there was around a 35% increase in coleoptile length simply by replacing RHT1 or 2 with RHT18. We moved this work to the field. We've developed some really clever systems for deep sowing down to 15 centimetres, much deeper than the recommended depth for Green Revolution RHT1 and 2 wheats, which are around, well, a grower once told me, no shallower than a matchbox is wide and no deeper than a matchbox is long, so 3 to 5 centimetres. We've taken our germplasm, tested them across the country. I think we now have around 40 site years of data where we've sown at up to 140 millimetres and shown quite clearly the relationship between coleoptile length and plant emergence with deep sowing and across a wide range of soil types. Here's a figure which just shows some field validation where we've contrasted maize, a commercial variety maize containing RHT2 with this RHT18 near isogenic sister line. And when we look more closely at maize seedlings that have emerged, digging those plants up, we saw a large number of unemerged seedlings associated with that very short coleoptile. In contrast to maize 18 sown at the same depth, where we had good emergence, we had a long subcrown internode, good crown node development, and very, very few unemerged plants. How valuable is this? Well, we've identified that across a range of studies, sowing early into moisture is absolutely critical, particularly as we move into longer season genetics. What I mean there is we don't want to risk developing wheats that can be sown late April, early May, at flower at the optimal time when they don't germinate for 6 weeks because they're dry sown. Because you delay flowering time past the optimal flowering window, you get a negative in terms of the reduced biomass with late emergence with dry sowing. Then you get another reduction in yield because of the delay in flowering. This is shown quite nicely here. This is some work from Walkery on a non-wetting sand where typically the soil beneath the soil surface is dry and there's good moisture. Some work from Bonnie Fleur. What Bonnie was able to show that with deep sowing of a long coleoptile wheat into moisture, there was up to 16 days difference in time to emergence in contrast to the dry, shallow sowing. And Bonnie, I don't show the yield data here, there was something like a 20% yield benefit with deep sowing. But if you look at the plot on the right, which is the long coleoptile maize sown shallow, it was delayed in emergence. And typically what we see is significant reduction in biomass and competitiveness. So significant increase in weeds. So another management issue for the grower. We wanted to do some work with some growers. And so we worked with a grower right on the eastern edge of the West Australian wheat belt, a place called Southern Cross. This grower here typically farms around 200 mils in crop rainfall a year. When we actually looked at the rainfall data for his farm, this graph here just highlights in blue post 2000 rainfall for each month versus pre 2000 rainfall. And what we typically saw was that over the past 20 years, significant reduction in sowing rainfall. And this was offset by a shift in summer rainfall. So these monsoonal troughs that come through the eastern part of the West Australian wheat belt was delivering significant water. But by the time Callum was going to sow his crop in April, May, that moisture had receded and was too deep for him to use. He had a full profile. He had yield potential of 2 to 2.5 tonne, but he couldn't take advantage of that early moisture because coleoptiles were too short. So he ran an experiment on his farm. He modified his planter. He had his father out there helping him. He invited the local growers and he sowed our long coleoptile wheats at 120 millimetres into rainfall. And he sowed their near isogenic sisters at the same depth. You can see here, Mace 18 at 120 mils, very good plant number, good establishment, and long coleoptile in contrast to the short coleoptile maize sown at the same depth, no plants. The crop on the left shows the benefits of deep sowing into moisture and how the delay in germination does delay biomass. Interestingly, Callum also through the season characterised growth, and what he was able to show, something we were not expecting, by germinating 6 weeks earlier, we had 6 weeks additional root growth. Because Callum was practicing a really good fallow and summer management of moisture, he had another 40 centimetres of soil moisture that he wouldn't have otherwise had access to if he had with the later germination. So roots grow 1.1 centimetres per day between germination and flowering. So that extra growth gives you the capacity to access deep moisture. And as I pointed to you before, with delayed sowing, you get the double whammy of reduced biomass, also later flowering. All in all, Callum on Twitter showed that in a decile 1 year with 76 mils of in-crop rainfall, he had record water use efficiencies. Well, I think they're overestimated because he didn't take into account that deep subsoil moisture. The benefit of early sowing was shown through more spikes per square metre and earlier maturation to avoid terminal heat and drought. And what Callum said for his environment, where typically it was a 1 in 3 success, he was getting at least 2 and 3 out of 3 years of success by moving to taking advantage of that early moisture. WA is the focus of our work at the moment, largely because of the problematic change in soil moisture. They're also, WA are very big into soil amelioration, spading, basically turning their soil over every 2 to 3 years to bury herbicide-resistant weeds, to bury waxy, non-wetting soils, and they use something called spading. Spading turns the soil over 30 centimetres, so it's nice and soft and fluffy, but you can't use it because it's too fluffy. You plant into it, your sowing depth varies anywhere from 2 to 15 centimetres, and with short coleoptile wheats, there's just not the rigour in the soil to ensure a reasonable sowing depth. However, using long coleoptile wheats, you can take into account or you can manage that variability in sowing depth. And so Steve Davies at Deepherd has been undertaking trials with our long coleoptile wheats and just showing that where in the past growers were losing a year with spading because of poor emergence and risking greater erosion because there's no ground cover, in one pass a grower can spade and then sow with long coleoptile wheat and pick up that season while protecting their soil because you've got reliable germination and ground cover. We've also, I guess, a breeder once told me, you develop a trait and you send it out and you target the trait and what it will deliver. But sometimes you get other benefits. Sometimes you get the opposite too. Bare patch is a superficial disease problem globally. It's been a holy grail, I guess, for pathologists and geneticists to develop resistance or tolerance to rhizoctonia. This is typical of what happens with bare patch. It's fairly infrequent and very difficult to select. David Roget at Sardi told me once that why don't we just sow big seed, sow them deeper, because if we sow below the inoculum layer, we avoid uptake and damage by the seminal roots. So David has since passed on, but I guess he'd be delighted to learn that the long coleoptile, when sown in these bare patch areas, we're seeing benefit in terms of improved root growth and avoidance or escape of rhizoctonia. And we're now working with GRDC and Deepherd to test this more fully, but not only for rhizoctonia, but also for other superficial pathogens. So this may be a way to reduce the impacts around the variability and the pathogenicity of rhizoctonia. Just following the coleoptile work, we've undertaken a range of studies across the country, both with breeding companies and ourselves. We've then modelled the value with deep sowing, both in current and future climates. And what we've established that moving into the future across Australia, deep sowing will provide, through improved water productivity and improved performance with high temperature, around a 20% yield benefit going forward. It won't always give you a yield benefit if you don't sow deep, if you've got good surface moisture. But on average, we're seeing significant benefit. And we're realising that in some of our own studies. We're also moving the technology into other crops. So we're working with oat breeders to improve coleoptile and mesocotyl length. And we're also working with canola breeding companies to try and improve the hypocotyl length. And there's genetic variation out there that's associated with increasing hypocotyl length to increase emergence with deep sowing. And we're releasing varieties. So we don't mean to release varieties, but sometimes the genetics just tends to hit the right buttons. And so working with breeding companies to move our germplasm with the right quality and disease into the hands of growers via breeding programs. The second trait is really around systems resilience, is around rapid leaf area and biomass accumulation. And I talk about it as vegetative, but I really think about it as early vigor and flexibility for growers in large sowing programs, or where there's issues with frost or with late breaking rains, to move into late sowing. And I talk to my colleagues in Canada who say, well, all their wheats are late sown. They don't grow so many semi-dwarfs because they rely on the vigor with the tall wheats to build biomass. We think we can improve harvest index with height reduction with alternative dwarfing genes, but by building biomass through genetics. And in undertaking this work, we've understood that there are many other opportunities in a systems resilience context with building capacity for late sown wheats around use of non-specific herbicides to control herbicide-resistant weeds, to manage waterlogging, sowing after the waterlogging event, frost and double cropping. But currently in Australia, there are very few opportunities other than barley. And we know that barley, while a magnificent important crop, Hannah, it's also doesn't have quite the same price premium as wheat, and provides a systems option. This is an example of late sowing. What this is a farmer's paddock. One side of the road, the farmer risked dry sowing, had rain the next day. You see the biomass in the variety calibre. This is on the South Australian mallee. And then the late sown, this is now sown 6 weeks later, very little biomass. And the crop flowered much later. So any grain that was set is quite pinched and small owing to the high temperatures and drought through grain filling. We've simulated the yield benefit with high vigour versus normal versus current vigour in current varieties. And right across the Australian wheat belt, we see that capacity to build high vigour and development of late sown varieties does have merit pretty much right across the country. So where did we get the initiative, the interest in early vigour? Well, it really was actually based on our understanding of multiple species. And in wheat, in water-limited environments, barley is the king and the queen of water-limited performance. And a number of reasons why barley does so well, but a key driver is its capacity to synchronise growth around key availability, key stages of moisture availability. And the key stage that I'm focusing on here is really around early growth. You can see here, side by side, you've got wheat and barley. You see the large difference in early vigour. And this is very typical of barley, much more vigour, which means that there's much less of the soil exposed. So you have much less soil evaporation. More water is available for transpiration to improve crop water use efficiency, but also to suppress weeds, but also to grow faster when you sow late. So what's special about barley? Well, barley has a large embryo and it has a very thin leaf. So for the same amount of carbon, it can spread its leaf area further. So those two factors together contribute significantly to the high vigour of barley. But when you look in wheat, they lack a large embryo and they tend to have very thick and small leaves. So we undertook some work to better understand what is the capacity to bring in new genetics for high early vigour, for water use efficiency, but also for late sowing. And in order to do this, we required some understanding of particular quantitative genetic parameters that would lend to improved selection, direct or correlated response. We found that narrow sense heritability for mean leaf width in this population was high for leaf width as a measure of early vigour, and it was much higher than numbers of leaves, for example. The genetic correlation of mean leaf width together with its high heritability translated to a high relative genetic gain when we looked at the correlated response for leaf area. So mean leaf width is a very simple trait to measure. You can eyeball it. As long as you standardise on seed size and seed from the same maternal environment, it can be screened very, very rapidly, very quickly. We identified 28 lines of breeding varieties across the world, which were known to be unrelated genetically and have high early vigour. We brought those into Australia, and we then undertook a recurrent selection program where we aimed to bring together important alleles for high early vigour through a recurrent process of accumulating favourable alleles and in replacing poor value alleles. This is demonstrated here, where with cycles of selection, we're improving trait value over time towards the development of elite lines, which then become the germplasm base for our breeding work. It's parent building for vigour. And we used S12 full SIB recurrent selection to sort of help drive our genetic gain, focusing on those aspects of breeding value, which will contribute to selection response. A very simple recurrent selection methodology with very, very large population sizes, try and reduce the impact of genetic drift to 6,000 lines per cycle, and we maintain pedigrees. This is what our breeding nursery looks like. Oops. So trays, augmented checks, and we would select. A student in 2015 took lines from each cycle, grew them in the same environment, seed sized them, and then showed quite nicely across a range of times of sowing, with and without nitrogen, a fairly strong linear increase in mean leaf width, and showing there the difference between cycle 0 in terms of plant growth and cycle 6. So we were quite successful in this phenotypic recurrent selection. We looked at embryo width and specific leaf area. While they weren't targeted traits, they were associated with this increase in leaf width over cycles. So the two drivers we saw in barley were coming through in selection for mean leaf width. Here's some of our material in the field being delivered to breeding programs. On the left, we've got a particular line, same sowing density, highly vigor selected, contrasting that with one of our more vigorous commercial varieties on the right. And as I said, there's always some benefits, or sometimes benefits. And I guess the other benefit we see with breeding for highly vigor is not only its capacity to build biomass with late sowing or improving water productivity, strong interest nationally, internationally, in the weed suppressive capacity of these high vigor wheats. It's probably hard to tell, but those leaves on that line on the right there, which is basically a wild catch-and derivative, about twice the width all the way through crop growth, which is linked largely linked to its capacity to suppress ryegrass and other weeds. There's also some element of increased root growth associated with shoot vigor. Roots and shoots are allometric in terms of biomass early, and so you're getting the benefit of suppression through limited light resource, but also below ground resource. The third trait, moving towards finishing, is a flowering trait. Again, around systems resilience and around giving growers the opportunity to build in that opportunistic cropping. And I guess as a GRDC member for years, we go and visit these frosted trial sites through WA. The growers had no sheep, and they would say, what do we do with our frosted crop? Because they're valueless. And so we would talk as a panel, and I asked them about awnless wheats, and they hadn't seen awnless wheats before. And they said, well, the value with awnless wheats is if you have a crop that is frosted, it can be cut and baled for hay. And wheat and hay has a high fructan content, so it's a prized commodity in terms of animal feed. So growers were looking for an awnless wheat variety, but if the season was dry, if it was frosted, they could cut for hay. If the season was dry, they could cut for hay. Or if prices for hay were high relative to grain, cutting the crop at flowering removes the risk of getting a crop through to maturity. It also removes the risk of reduced grain prices. If grain prices are high, you can sell this grain. So we went back, looked at our old germplasm, and we identified awnless wheats that had high premium grain quality that were also awnless. We did some work on that material and established that the awnless wheats have no penalty in terms of yield, except in extreme environments. So this was a trait that we thought was of value. We made the genetics, this is to show you what frost looks like through West Australia. Frost damage at flowering, and the value to the grains industry is between $400 and $800 million a year. So this is some of the genetics we developed. These are near isogenic pairs with and without the awn trait, and just highlights that there is capacity to build genetics that can be used for building a new industry around wheat and hay. So
any of these traits from
no. Oh, one of the great things about university, isn't it? Yeah. So we released 2 wheats. We actually never went to release these as varieties. The breeding companies said, no, we need to release them because there's nothing in the marketplace. So they've taken the genetics, they've released material, and they now have germplasm coming through their programs with improved disease, improved quality, improved yield potential. But we've developed an industry worth about $1 billion a year, particularly for growers in frost-prone areas, giving them the capacity to have a crop that can be harvested as grain or sold as hay. So in conclusion, I guess I hope I've made the point that I think breeding rain-fed crops for future climates will be incredibly challenging for a number of reasons: the genetics, the signal, and that I think there is significant opportunity, given the variability from season to season and within season, that growers can be given the tools to build in their systems knowledge and build in their choice of genetics around what will permit improved profitability for them. It doesn't mean that we don't still focus on managing our soils, managing our soil moisture, monitoring our crops any less. We need that information. But with that information and with the genetic options and their farming system, we allow the growers to play the season and give them the capacity to reduce production risk while maintaining high financial return at low environmental risk. Novel genetic variations being targeted and assessed. I don't think anybody else in the country or even globally is looking at the genetics we've been developing. There's interest in the genetics, and this is only a small snapshot of what other genetics may be possible in this system's resilience. And I also just want to highlight how important it is that in delivering your research and delivering impact, you maintain very close links with the commercial breeders, because the breeders are the ones that take the germplasm up. They're the ones that fast track your genetics through their pipelines. And as I said, in some cases, it results in joint releases. Other cases, it allows breeders to provide material for population development. It also provides two-way dialogue around what's working and what's not working in your pre-breeding activity, but also some other insights that you may not be aware of. So I thought to finish up, because the involvement with students is such an important part of this program, I just thought, for the hell of it, I thought I'd just highlight 3 lessons since leaving UQ for me as a scientist. And I think of my career as being an undergraduate in horticulture, a master's in a wild species, wild mung bean, PhD in biochemical genetics in soybean, and then postdoc and career now in abiotic stress in wheat. And none of those different stretches built on each other. They were disruptive in time, often in location as well. But one thing that drove me the whole time was curiosity. I go to bed thinking about my science, and I wake up thinking about my science. It's what drives me. It's what gets me through the difficult times at work. And I've got a very, very supportive wife and family too. But be curious and be excited. You know, it's such a wonderful part of our science. And I guess what drives us. None of the ideas I presented are my own. I got them from farmers. I have quotes throughout my talks on things that growers have told me, things to look out for. Growers are the best, they're the best scientists. They're very keen observers, have long memories. They tell you things. It's you as a scientist who's responsible for picking those up and interpreting them, and then in some cases then testing. The coleoptile work, the early vigor work, the weed suppression work, late sown wheat work, the awnless work, were all commentaries from growers in providing growers with the resilience to maintain profitable enterprises in very challenging and very dynamic rain-fed systems. So attend grower field days and talk to growers. They're amazing sources of knowledge. And the final one I think too is I've been lucky that I've been able to work across multidisciplinary teams. And you don't always work with those same individuals or those same groups to the same extent year in, year out, but you pick from each other, you learn. The knowledge base is so critical. And I think it stops you from getting stale. So engage with statisticians, engage with physiologists, pathologists, molecular biologists, agronomists. With that, some acknowledgements and any questions?
Thanks very much, Greg. That was really good. Thank you very much. And it's really good too that you left time for questions, because sometimes our speakers are so filled in with their own importance that they don't actually think about that. So we do have plenty of time for questions. You're happy to fend for yourself? Can I start with one?
Please. Oh, please.
When you talked about getting all those different RHT genes in from all over the world, now all of them were from mutation?
Yeah, they were
Program?
out of mutation programs. Yes.
Okay, and were they different novel loci, or were they allelic variants of known?
So we actually brought in around 25. At the time, we were building the perfect RHT1 and 2 markers, and when we actually screened the germplasm, before we really pushed the GA screening, we identified that probably a third were RHT1 or 2 mutants. So they were not novel at all. They'd been misclassified, which didn't surprise us, I guess. In terms of the other genes, I think they're largely independent. There are 3 genes which sit on chromosome 6A, RHT 16, 18, and 24, but 2 are dominant and 1's recessive. So we know that they could be allelic. They could be allelic, but they're so close together, it's unclear. We're working more closely at that interval to try and break them apart. So they may be the same gene, but different alleles at the same locus. Right. So yeah, but I'm working with Eloise Fu and others to better characterize the metabolism and the GA biosynthesis pathway and how these genes regulate plant height. Some of them are GA deficient, some become GA insensitive through time. So the different genes reflect, in many cases, different GA response types.
Right. So, yeah. Okay. And can I get you to repeat the question, because the people online won't have heard the question?
That young fellow at the back there.
Questions about the early vigor study. And okay, you said that early vigor results in thinner, wider leaves early in growth. First question is, does that correlate to thinner leaves when the canopy matures? But really my question is about water use. So what is the interaction of the greater leaf area early on water use, and then subsequently when the canopy matures, does that affect the early use too? I mean, you might not have a chance to measure water use.
We know SLA is maintained through ontogeny, through canopy growth. We've not done detailed water use studies, but we do know that based on water balance work, we know that having that greater vigor early, when large part of the rainfall in the Australian wheat belt occurs, that we're able to reduce soil evaporation from up to 60% down to about 25%. So in terms of water balance, having that water saved by reducing soil evaporation has been significant through that greater leaf area early. And sorry, I should have reiterated the question. So it was around specific leaf area and water balance around changing SLA through crop life. But yeah, no, Chris. Yeah, they stand out. They're very pale green in the field. The lines stand out quite clearly all the way through growth. If there is an issue with them, when we go extreme SLA, they are a little more predisposed to herbicide damage. So we've got to manage that aspect. But in the back cross-derived lines and top cross-derived lines we've given to the breeding programs, those lines are often removed because they're unwieldy. They tend to lodge as well, because not only is the leaf thinner, but the leaf sheath which holds the stem is also thinner, and so it's more vulnerable to lodging. So, yeah.
It's a question from Eric.
Yeah, it's not a related question. I mean, first, this question would be a comment. I think the SLA will be consequence of the bigger. But my question is, this is very bigger. With your widening area, widening width, does it not affect your tillering, and therefore give the fish actually less or bigger important before tilling?
So Eric's question was, is early vigor and leaf width particularly, does that reduce tillering? So we're not aware of a change in phylochron interval. What changes tillering in wheat is leaf length. And so we put a lot of effort in ensuring that in breeding for wider leaves, we maintain a shorter leaf. Wheat does not produce a tiller until the previous leaf, initiate to produce a tiller, the previous leaf is fully elongated, unlike barley. So leaf length rather than leaf width is the main driver of tillering in wheat. Yep. Any coleoptile questions?
Doing PhD to get sort of embark on their careers like you. So you gave us 3 really good examples of approaches that work. Have you worked on any trait targets where you put in as much effort but it didn't work? And how would you encourage people to keep going on that?
Yeah, talking to the students this morning, I talked about 30 years and you have your foci. I guess that's what keeps you excited. What keeps you excited are the little sidetracks. And so I've had other traits. I've had rate of grain filling. So the rate at which grains fill as a trait. I've had radiation use efficiency by developing wheats that lack a ligule. So they're very, very erect. So you could squeeze more plants in and maintain higher yields with a more efficient radiation use. And there have been other things as well, root architecture. I guess, Mark, if you believe in something, you pursue it. And if what you believe in doesn't follow dogma, dogma's not right. So you modify your model. There aren't any traits I don't firmly believe in, but there are traits that do require much more work. Root architecture is a case in point. The challenges of root architecture, despite the importance of it for accessing deep moisture and deep nitrogen, it's an incredibly challenging trait because of, I guess, the variability, the stochastic nature of soils and soil biopores. The other trait that I've worked hard at, and it's getting some traction, is the rate of grain filling, where for the same duration of ear growth, there are wheats there that will accumulate grains at 25, 30% higher grain rate. In a terminal drought or high temperature finish, that would be an incredibly important trait. The problem is it's very difficult to measure. And so while I provide germplasm to the breeding programs that have the trait, they lose it very quickly because they have no valuable selection tool currently. A genomic prediction and other tools may lend to that trait. But the traits in their delivery and their adoption have to be reliably measured. Value proposition has been demonstrated, germplasm has been delivered, but very hard to select. And so there are a couple of examples. So, but I don't know whether it's by chance. I think the traits that have done well are early traits. They're traits which are less masked by environment, and so they're probably a little easier. They can be done in a glass house. They can be translated and validated in the field readily. So it might be why I've seen less issues with them, because the issues have been addressed very, very early before I got found out in the field. So, Anthony?
Perhaps a big substance either I've seen one or actually two, and they're alternative talking gene sister lines. It seemed fairly consistent that sister lines canopy wasn't dense. I'm just wondering if there is an effect on tillering, reducing tillering in the sister line.
That's it may have just been because that photo was taken the first year of our commercial seed increase. So it was under irrigation at Condobolin. So that may be there's plentiful commercial seed, but not of the sister lines. So this is the first year we had them grown out. But I guess I wanted to, and we know since that time, that was 2018, I think 2019. This material has been used across the country. One of those lines has been considered currently as a commercial variety in its own right, and has the same yield potential deep or shallow sown. So we know there's no trade-off with with yield and potential quality. But for all intents and purposes, they look very, very, very similar. in the field, and that's reflected in their yield components and their performance. But good observation.
I've got lots more questions I could ask, but I'm going to try and encourage someone under the age of 50 to potentially ask a question, and maybe even not being male as well. And all of the females look at their lap. Oh dear. No eye contact.
Andy Borrell always has a question.
He's not under 50.
Oh, oh, okay.
No, go on, Andy, then.
Just looking at the, you say the base plus the RHTA replace RHT.
I'm sorry, yeah, we've replaced the GA insensitive with RH18, yeah.
So it's just following that, being quite similar to that question with the RHTA, getting the same about sort of
Yeah. If anything, on par, RH18 is associated with probably a 2 to 3% further reduction in height. One thing I haven't talked about, and I guess that'll be my next talk, we're seeing wheats globally sort of reaching a plateau in terms of harvest index, which is largely flowering time and height. And it's using single dwarfs. There's capacity to develop double dwarfs, RHT1 and 2, but they're too extreme and they're biomass limited because of the effects on cell growth. We have near isogenic lines now, which are combinations of the green revolution and the GA sensitive dwarfing genes. We call them sesquidwarfs because they're not as extreme in height reduction, but they are short enough that you do increase harvest index even further, but without compromising biomass, because you're not displacing the biomass. You're limited to the RHT1 or 2 biomass, but not to the extent you would with a more extreme dwarf. So there's some interest in some of those genetics in the breeding programs, and the breeders are actually, with the markers, are enriching for those combinations of those weaker double dwarfs. So something quite novel that may come out of this because they're non-allelic genes and because of the physiology lends to these weaker double dwarfs with the high yield potential.
We have time for one more quick question if anyone wants to go for it.
Which one, sorry?
If it holds profit with nutritional goals, is it the same for a public program, plants in bigger cities?
Yeah, it's a good question. I mean, the other way we can improve seedling vigor is by increasing seed size. Absolutely. Growers are reluctant to do that because of the cost. And so where we've undertaken studies where we've increased seed size by 20, 30%, but while maintaining the same grain number, we see the same benefit in terms of early vigor. So if I'm reading your question correctly around seed size, we believe that having the genetics in the seed for any seed size is probably going to be more valuable to a grower, particularly given that the seed that growers use is often seed harvested from their own farms and often seed that's harvested under hot, dry finishes. So you may not get the potential for large grain in many environments. So, but if possible, I mean, we've talked to breeding companies about seed nurseries, irrigated seed nurseries where they could harvest large seed, but growers or breeders don't see the value proposition in that. So yeah, so rather than focus on large seed, we're focusing on having the genetics for early vigor independent of the seed size. Have I answered your question? If not, I'll be around. Thank you.
I can also hear some growers saying, oh yeah, seed companies would love to increase seed size because they'd charge me the same for a bag, but I'd get 20% less seed for sowing. Am I right? Well, I mean, thanks very much, Greg. That was really good, and thank you very much also for focusing on things that you're excited about and helping other people to see that if you can keep that excitement, you can have a fun career. And on top of that, if you keep that excitement and be curious and start to deliver things, that's great as well, because Greg talks about going and talking to growers. It's a lot easier to go and talk to growers when they already know about some of the stuff that you've done. And that's something that's really hard as an early career researcher. They don't know anything about you. And you'll come up with some ideas. I mean, I know myself, I used to go to grower days and I'd talk to growers and I'd say, oh, what about this? And then, you know, some of them would be, you know, very polite and some of them would say, you're an idiot. I mean, and it's true, because every single idea we come up with, 90% of them are stupid, right?
But they're thrilled that you've made the effort.
That's true. That's
They
very
really
true.
are. And because many of our projects are funded by growers through
Yeah.
levies, they want to see you out there learning and understanding their system.
Yeah.
So they'll tolerate your views
For a few years.
for a few years.
If you don't change your views.
Yes.
Okay, well, I'll just ask you to join me again in thanking Greg.